Hi, my name is Ruchira Singh, and I am the Dean's Professor of Ophthalmology at the University of Rochester. Today, I'm going to be talking about vision loss in CLN3 disease and looking at providing more information about the current knowledge on the retina disease phenotype, as well as the utility of organoid models to be able to study the retina disease pathology in CLN3 disease. So let's get started. So the first thing that I'm going to be talking about is the vision loss phenotype and what is known about it in classic CLN3 disease. And so in classic CLN3 disease, all patients that are affected by the disease have vision loss. So it occurs in everybody. And it is the hallmark of the disease and the first clinical symptom in most patients, about over 80% of the patients. And it typically occurs between 4 to 8 years of age, like the start of visual impairment is seen around 4 to 8 years of age. It can happen earlier, it can happen later, but the mean age of onset is around 6 years. And it very, very quickly progresses. It shows a very rapid progression and leading to legal blindness in late childhood and early adolescence by around 12 years of age. And there is widespread retina degeneration. So, vision loss in CLN3 disease is because of retina degeneration, and the retina degeneration is not limited to a specific cell layer, but you see it all across the retina, affecting both the inner and the outer retina. And the clinical phenotype can mimic other common forms of retina degeneration, especially early on. And it is and if you don't have a family history of the disease, the ophthalmologist would be more inclined to maybe think you have Stargardt's disease or retinitis pigmentosa, given the really early onset of the disease. And because some aspects of CLN3 disease, retina pathology seem to mimic these more common retinal degenerative diseases. So, CLN3 disease, similar to the classic CLN3 disease, vision loss is also seen in all patients with the protracted CLN3 disease. And again, it's the first clinical symptom in the protracted CLN3 disease patient group too. And the age of onset is around 5 to 9 years of age, with a mean age of onset, again, between 6 to 7 years. So between the protracted disease and the classic CLN3 disease, there is normally not a big difference at the age of onset. However, the other neurological symptoms in the protracted disease do not develop till very late in the disease. So, but the classic CLN3 disease and protracted disease in most patients, although there are reported cases where vision loss occurs later, in most of them it occurs in early childhood and progresses very rapidly and again leads to legal blindness. It has, similar to the classic form of the disease, it leads to widespread retina degeneration. Again, it's not restricted to any specific part of the retina. And again, mimics the other common forms of retina degeneration and can be misdiagnosed, just like the classic CLN3 disease. Then comes, and then there is another form of vision loss associated with CLN3 mutations, and that is in the non-syndromic isolated retina degeneration. So a subset of patients that have mutations in CLN3 gene, they develop an isolated retina degeneration phenotype, and this is the non-syndromic form of the disease. And it generally typically occurs between the second to fourth decade of life. So the visual impairment starts in the second to fourth decade of life. And there are two different phenotypes: people that develop it that start early around the second decade of life, and those that develop it later. And they show a slightly different phenotype but when you compare it to the classic CLN3 disease or the protracted CLN3 disease, this disease progresses slowly. However, it also leads to legal blindness. And again, by fourth to seventh decade of life. And it is also due to retina degeneration and can mimic other common forms of retina degeneration. However, unlike the CLN3 disease, the syndromic CLN3 disease, in the non-syndromic isolated retina degeneration, it's the outer retina, the photoreceptor retinal pigment epithelium RPE, photoreceptor RPE complex that is affected, and patients mostly show a rod cone dystrophy or a macular atrophy phenotype. And it's mostly associated with homozygous point mutations in CLN3 gene, but the genotype phenotype variability of CLN3 disease is a challenge, and you cannot just say this group of patients will only develop isolated retina degeneration, because there have been instances in the literature where people have later on developed a neurological phenotype and they were diagnosed with just isolated retina degeneration. So the genotype phenotype correlation isn't very well established for CLN3 disease. The key takeaway point here is that all disease causing mutations in CLN3 gene, whether they lead to the syndromic form of the disease or non syndromic form of the disease, lead to retina degeneration. And in the syndromic form of the disease, there is widespread degeneration of the retina that affects the whole retina, both the inner and the outer retina, whereas in the retina-restricted non-syndromic disease, it's the outer retina, the photoreceptor retinal pigment epithelium complex that is affected. And again, this highlights the importance of CLN3 gene for retinal homeostasis, because the retina, especially the outer retina, is affected by every disease-causing mutation in CLN3 gene. So let's look a little bit more into the retinal degeneration in CLN3 disease and maybe compare it to the more common macular degeneration and retinitis pigmentosa phenotype. Based on the extensive characterization of the disease in the human eye progressing through the disease, it's been shown that the earliest pathological changes are seen at the photoreceptor RPE complex. And it's at least in the syndromic disease, all the cell layers seem to be affected more or less at the same time. But if you were to having access to patients that have really early disease, and sometimes being able to look at the sibling of a patient that had been diagnosed before and also carries the disease helps figure out the earliest pathology in the disease. And it's been reported that the earliest change occurs in the photoreceptor RPE interface. And this happens in both syndromic and non-syndromic. However, the inner retina is highly involved in the syndromic form of the disease and very early involvement too. And the disease initiates at the photoreceptor outer segments and then progresses gradually inwards. And retina degeneration in CLN3 disease follows has some similarity to macular degeneration and retinitis pigmentosa, which are common retinal diseases, although retinitis pigmentosa has a very variable profile and very heterogeneous. There is photoreceptor loss, there's progressive vision loss due to retina degeneration. There is photoreceptor and RPE dysfunction, and one or the other cell type or both cell types can show atrophy. There is changes in the lipofuscin or autofluorescence levels in the RPE monolayer, although they might be different, but increased in macular degeneration versus decreased in CLN3 disease, or in some forms of retinitis pigmentosa, like Morticare-associated retinitis pigmentosa, you might see more of the phenotype that's seen with the CLN3 disease. So it does seem to mimic certain aspects of the more common retinal degenerative diseases. And for example, in macular degeneration, it's the central retina or the macula that's affected, and that's also what's initially early on affected in CLN3 disease. So there are a lot of similarities, but mechanistically, it is unclear how similar these diseases are in terms of disease development and progression. So there have been multiple approaches that have been used to study the retina pathology in CLN3 associated disease. One of it is studying the disease progression longitudinally in the patient's own eyes over time. So here in the left, if you can see that it's basically using non-invasive retina imaging and retina imaging modalities and functional testing like electroretinography are pretty advanced. So they are able to tell you the function of specific cell layers in the retina, and you're able to, over time, see the progression of the disease. So he has just mentioned the fundus photo, OCT, and ERG analysis that allow you to look at the structure and the function of the retina in the patient progressing through the disease. And on the right is showing another way is to look at the histopathology of the human donor eye. However, this is limited to seeing changes that are occurring in the late stage disease or the end stage disease when the retina has completely degenerated, but it still is a valuable tool to see what is happening in the human eye that has CLN3 disease. So, although these, although there are advantages of using uh, imaging studies on human patients to understand the time course or the sequence of pathological changes that are happening, it it can give you some clues into the mechanism or the cellular basis of how the disease is developing. And this is very important information to be then able to follow up and see in the lab, like, how which cell type is affected and what could be happening to it. However, there are limitations in terms of most kids are identified with the disease, have already had a significant amount of visual impairment, and the retina has already showing a lot of structural changes, and it's then becomes difficult to really isolate which cell type in the retina or which part of the retina is affected. So there are some challenges, but this is a very good approach to figure out the earliest pathological changes in CLN3 disease in the patient's own eyes. And then another approach that is being used now for several years is animal models of CLN3 disease. So mouse models or rodent models are very often utilized to study retinal diseases. They're not very good at capturing the disease pathology of diseases that affect the macula. And this is just because rodents don't have a macula or a macula-like structure. And but for other diseases where the macula isn't important, or where there are several retinal diseases where the studying mouse models of inherited retinal diseases has led to the pathology and understanding of the disease. However, for CLN3 disease, there have been several mouse models, both the Delta 7-8 and the knockout mice, and on multiple strains. And none of them show really the ability, none of these existing mouse models thus far have captured the human disease phenotype. And this is particularly relevant when you think about it from the perspective of the retina. These mouse models have been very limited in capturing the retina disease phenotype. And more recently, to overcome this, there's been there's a transgenic large animal model of CLN3 disease, the CLN3 mini swine model. And you can see here that there is retina degeneration or photoreceptor outer segment degeneration already at 1 month of age. So this is a very, very early onset degeneration of photoreceptor cells, and very similar to the humans, the earliest pathology occurs at the photoreceptor outer segments. And the disease model goes over to far. In the pig retina, there is extensive retina thinning and involvement of other cell layers. You can see the abnormal PNA lectin staining here in the other cell types. So there is a very good recapitulation or replication of the retina disease pathology early on in the CLN3 pig model, and it's very localized to the area centralis, which is again which is the equivalent of the human macula. So it makes it a very good preclinical model for being able to do therapeutic testing. However, again, coming back to the mouse model, there is some pathology in the mouse model that's been reported, and it's mostly limited to some changes in the inner retinal layers. The photoreceptor cells are not affected in the mouse model. And so, although CLN3 mutations can lead to some phenotype in the mouse models, as of yet, they do not seem to recapitulate important aspects of the human disease pathology. So other approach and very routinely utilized approach of studying the mechanism underlying retina degeneration, or the more common defects in CLN3 disease, the molecular basis of it has been the use of cell culture models, and specifically the retinal pigment epithelium, RPE cell models. And till very recently, till the advent of the iPSC technology, or the pluripotent stem cell technology, this was limited to using immortalized RPE cell lines, which include the CLN, knocking CLN3 out in ARP19 cells. And a few years ago, or like right now, another approach that's been used or published is using siRNA to knock out CLN3 in RPE1 cells. And again, these are both immortalized cell lines, and they have improved our understanding of the molecular mechanisms. However, they're very limited in mimicking important disease phenotype, and several studies have shown a big difference in the transcriptomics and even like formation of tight junctions between these immortalized cell lines and the human RPE in vivo. And this has had a significant effect, right? Because important disease processes that are affected in CLN3 disease, like phagocytosis of outer segments, are difficult to replicate in immortalized RPE cell lines. Although, yeah, they have truly been helped us understand the differences between normal versus CLN3 mutation carrying cells in terms of specific disease, specific cellular processes. So, as I said, once iPSC technology has enabled the development of RPE from patients' own cells, and these are being utilized to study the disease pathobiology too. And this includes both the use of patient-derived iPSC lines that have the most common mutation, and then the gene-edited isogenic line where a mutation was introduced into normal cells, so that it helps you isolate just the impact of the mutation in the absence of any other background differences. So these two approaches have now been utilized to link CLN3 mutation to specific defects or specific pathology associated with CLN3 disease in the RPE cells. And so they provide another platform to be able to study the CLN3 pathobiology in the retina. And so more recently, we've developed the iPSC retina cell model. And again, these have utilized from the iPSC derived iPSC derived cells from patients that have the most common deletion mutation spanning exon 7 and 8, and both using patient derived cells as well as gene edited isogenic lines. And so what I'm trying to show here is that, and I will go into this in the next few slides, is that this approach allows you to have the retina with, so you can see normal retina and normal RPE, and you can see normal retina with, sorry, mutant retina with the mutant RPE. So this is basically a comprehensive model of the human retina, or a human retina cell model of the disease. And I'm going to go into more into how this is developed. So in the iPSC, and then the pluripotent stem cell derived model of retinogenesis, the retina develops differently, or the retinogenesis occurs slightly differently than how it happens in vivo, in that the neural retina, which includes the photoreceptors and all the other retina cell types, neuronal cell types, and the support epithelium, the retinal pigment epithelium, are separate entities. They don't co-develop. And this actually is a boon in a way, like that it has enabled the ability to be able to study the retina organized versus RPE independent of each other, and see what is the independent effect of disease causing CLM3 mutation on the retina, neural retina, versus the RPE cell layer. So here you can see this is the neural retina, and these are the outer segments, and this is the RPE monolayer. And this is a higher magnification, basically showing you the outer nuclear layer in the dish, and you can see the outer segments. And so this is the retina organoid. So again, it enables studying them as individual entities. So, however, having them as individual entities does not help resolve the need of having a comprehensive retina, because when you want to do therapeutic testing, or you want to identify a disease mechanism or validate it, you would want to see how the whole tissue is behaving. So the comprehensive retina, where you can now combine the retina organoid with RPE, has been developed by my laboratory recently, and this allows the investigation of neural retina, or the retina organoid, versus RPE dysfunction now in promoting photoreceptor cell loss in the whole combined tissue. And so basically, to be able to make the retina organoid RPE model, we use engineered extracellular matrix and some cues from in vivo development of the retina to promote the retina organoid and RPE to form a co-culture model where that is conducive to understanding the role of or studying the photoreceptor RPE interaction, which is where the CLM3 disease develops. So if you, this is showing the developed RORPE model, or the comprehensive retina model, in a pluripotent stem cell derived human cell model, so a human cell model of the retina. And what you can appreciate here is that now when you have the RPE, you actually see that these outer segments are facing towards the RPE monolayer that's below, and they're no longer mislocalized. So this actually provides a platform where you're able to get a more physiological neural retina too in a co-culture model with RPE. And this is showing phagocytosed outer segments by the RPE cells. So these are endogenously produce. So this is not like feeding photoreceptor outer segments. This is actually outer segments in the retina organoid that have been ingested by the RPEs. And this is a day 7 culture. So this tells you that this is physiologically happening over time, where the RPE is ingesting the shed photoreceptor outer segments. And this is again showing outer segments on top of the RPE, outer segments in the RPE, as you would expect it, and again showing the promotion of outer segments. And the also the RPE microvilli and the ingestion inside the RPE microvilli of photoreceptor outer segments. So from the perspective of being studying a disease that occurs at the photoreceptor RPE interface, this model provides a very unique platform and that is valuable for understanding what would happen uh, in a diseased tissue versus normal physiology. So coming to CLN3 disease and modeling this, you can see here that in the in the stem cell derived model, if you now don't have the RPE and you just have an isolated model of the retina organoid, you can see these outer segments, and these are about a year-old cultures. And the outer segments remain completely intact. Nothing happens to them in the absence of RPE. And you can see again here that they stain for rhodopsin and melalopsin, which are photoreceptor outer segment markers. And this is looking at apoptosis or cell death and tunnel with tunnel staining, and you can see here that there is really no difference in cell death between control versus mutant cultures. However, now if you restrict the CLN3 mutation to the RPE layer, that is independently sufficient to lead to uh, photoreceptor retina degeneration in the CLN3 disease. So if you look here, this is a control retina organoid cultured with a control RPE, and you can see abundant photoreceptor outer segments, rho positive and ML absent, so both rods and cones. Whereas now, if you have a total control retina organoid and mutant so here you have a control retina organoid, but you have put it together with a mutant RPE. So your retina organoid is normal, but your RPE is mutant. And in this scenario, you can see that there is almost complete loss of outer segments. And again, showing it in a different view, where you can see the outer segments and the RPE. You can see in the control retina, there's a lot of outer segments and within the RPE. But here you have a complete loss of outer segments in the retina. And this is mediated by just an RPE specific defect. So anyways, the model allows you to independently to look at the cell autonomous role of RPE in promoting neural retina or retina organoid degeneration in culture. So you can see here, the platform that we use was again, you have a complete control culture. And so you have the organoids that are controlled and the RPE that is controlled. But you can also mix and match. And this is very, very valuable for understanding the role of a specific cell type or a specific cell layer in promoting retina degeneration. And you can see here the RPE is mutant and the retina is controlled. And this is a complete mutant culture where both your organoid and your RPE are mutant. And now you compare the phenotype here. And you see in the control cultures, you have outer segments in both the retina organoid and being ingested by the RPE due to phagocytosis. And you have, again, in a different view. And if you can see here, you would see that in just having the mutant RPE, there's a degenerate, their outer segments are gone, and there's nothing for RPE to phagocytose anymore. And so finally, when you have the complete mutant cultures, you see again the same pathology, maybe a little more extensive, so again suggesting that a role of RPE dysfunction in promoting retina degeneration and perhaps the early onset of the disease in CLN of the retinal disease compared to the neurological disease in this. And so again, this is showing that the number of outer segments are not just reduced within the retina organoid. This is the quantitative analysis in the same model. And you can see that there are control organoids cultured with CLN3RP or the total mutant organoids have very little rhodopsin as well as very little cones positive for melalopsin within the retina organoid, but also within the RPE. So basically, it mimics the loss of outer segments in the retina and also within the RPE, but this is an RPE initiated effect. And that is something that would be very difficult to figure out in vivo. And the reason CLN3 disease for a very long time was considered to be a photoreceptor dysfunction for the loss of outer segments is because the thought was that the outer segments are lost and that it's a photoreceptor dysfunction, and then that's why the RPE is not phagocytosing. But this data here is showing that the RPE is actually dysfunctional, and that is leading to outer segment degeneration and consequently less phagocytosis by the RPE. So it's a very important tool to be able to study the disease pathophysiology in a patient relevant human cell model. And this is just emphasizing what I just showed you there, that just this is a human donor eye. And you see that complete loss of rhodopsin here. And this is normal human donor eye, and this is patient-derived human donor eye on the right. Right. And you can see in the patient donor eye, there's complete loss of outer segments, which is basically what is mimicking what's happening in the iPSC-derived cell model. And although the RPE is still there intact, it is it would be very difficult in vivo to figure out that a RPE dysfunction is promoting photoreceptor degeneration and photoreceptor outer segment loss. But the organoid model is a powerful technology to be able to answer this question. And also from the perspective of therapeutic, the organoid model allows us to test a potential therapeutic in a human model. So just showing an example here, we've been looking at recombinant acylceramidase as a treatment for CLN3 disease. And so we treat our mutant cultures. These are complete mutant cultures with recombinant acylceramidase. And you can see here in the untreated cells, there is very little presence of outer segment proteins. However, in the treated cells, now you can see that the rhodopsin in green and amylopsin in red are abundant. So you can see that we are able to rescue the phenotype in a human model of the disease. And this has a very, very high therapeutic relevance because you are actually treating the disease pathology. And so the mechanism, it validates the mechanistic defect by showing its ability to target the actual disease pathology in the patient's own cells. So to summarize, what I've shown today is that, or what I've talked about today is that all disease-causing mutations in CLN3 lead to retina degeneration. The loss of photoreceptor outer segments is the earliest disease pathology in CLN3 disease, and retina degeneration in CLN3 disease can mimic aspects of more common retinal degenerative diseases like retinitis pigmentosa and macular degeneration, but there are distinct pathological manifestations of each, and the molecular defects and mechanistic basis is likely to be very different. And there are several viable approaches for studying retina degeneration and CLN3 disease, and this includes human imaging studies, animal models, cell culture models, including immortalized cell lines and patient-derived retina cell models from the iPSC technology. And finally, what I showed was that patient-derived retina cell model of the CLN3 disease can capture an important disease pathology and the earliest disease pathology, which is basically the loss of outer segments and photoreceptor degeneration, and thus provides a very suitable human platform for both mechanism-based studies as well as for therapeutic studies. And with that, I would like to end with acknowledging my laboratory, especially Jimin, who did this, a lot of this work, as well as Sonal, who supported her, and all the other, I would also like to thank all the funding support I've had to be able to look at CLN3 disease in a human cell model. Thank you.